Uh, thanks. Thanks a lot, Supratim, and thanks a lot for inviting me to give this talk. I fully realize the sort of irony of being an online talk at ICTS while all of you are sort of sitting there, but I'm really sorry for the circumstances in which I had to give this. But be that as it may, I'm very excited to tell you about some of this work. So I'm going to talk about how we sort of discovered a few years ago that this idea of multiple stable states in in sort of communities could be explained using this idea in game theory called the stable marriage problem. And this story sort of starts somewhere where you maybe don't expect it to start, which is this guy over here, which I'm sure some of you are familiar with. Um, so basically, the idea is that in roughly the sort of 60s or 70s, a lot of detergent companies, and Tide was sort of the biggest of them, were sort of like really in the sort of radars of a lot of different regulatory bodies, especially in sort of North America. And the reason for this was that I don't know if you knew this, but so a long time ago, basically, like the primary component of detergents used to be phosphorus, and this used to be for sort of softening water. But sort of one big effect of including a ton of phosphorus in these detergents used to be these things over here. There used to be these gigantic cyanobacterial blooms that used to form all over towns. And so just to give you a perspective of what you're looking at over here, you're looking at this big lake called the Lake Erie, which is one of the Great Lakes in the US and Canada. And basically, this entire lake, not just this lake, but several other lakes, would get completely sort of flooded with these sort of blooms of cyanobacteria. And basically, the idea that people had at the time was that the cyanobacteria would bloom because really the limiting nutrient for their growth was phosphorus, and because there's a lot of runoff from detergents which contain phosphorus, these things would just go out of control. Now, as you imagine, detergent companies were not really ready to address this. They believed that phosphorus wasn't the key ingredient that was causing the cyanobacterial blooms because they'd done some of their own experiments in the lab, and that didn't seem to turn up something. And so, really, at this point, the Canadian government, especially, was kind of really, concerned. And so they recruited this guy, David Schindler, to do a pretty crazy experiment. And so the crazy experiment that sort of David Schindler did was that he was given access to these extremely pristine lakes. One of the examples of which you see here in sort of the north of Canada, and so he sort of embarked on doing this pretty insane experiment. So he would go to one of these clear lakes, and basically he had a pretty clear idea. He would go to these lakes and basically find two adjacent lakes that were next to each other. And to one of the lakes, he would add nitrogen, and one of the lakes, he would add phosphorus. The key idea being that kind of nitrogen was the contention that the detergent companies were making was the sort of key player in causing these cyanobacterial blooms. And so when he did some of these experiments, really what he found was the following. So here's an example, and actually what's now become quite a famous picture or a photograph taken from a helicopter of essentially two lakes that are very, very close to each other. And just to again give you a perspective, this over here, if you can see my cursor, is a little road that goes in in between these sort of two lakes. And sort of you can kind of very clearly see that on the left, this lake is turbid, and on the right, this lake is clear. Right? And maybe you don't get any points for guessing that these are actually lakes manipulated by Schindler himself. And to the left, in fact, he had added phosphorus, and to the right, he'd added nitrogen. And this kind of made it pretty clear that phosphorus really was a big culprit. And so this is about the turning point for this whole battle, where the Canadian government actually put in strong regulations in place for detergent companies to reduce the amount of phosphorus in them. And so the key idea that I wanted to take away from this story, which I still find really wild, is the idea that really you can take a natural ecosystem, add nutrients to it in some way, and really make it transition from one state to another. And really, this is not just any state to any other state. The idea really is that the clear state is really a stable state. You really need a big perturbation to go outside of the clear state. And indeed, the turbid state is also, in some sense, irreversible because once sort of communities entered this big cyanobacterial bloom state, it was sort of very hard for them to recover. And so the idea here is that natural ecosystems can be in different stable states, and that perturbations of some specific kind, maybe adding phosphorus but not necessarily adding nitrogen, can trigger transitions between these states. And this is not just a thing that happens in these big lake ecosystems, of course. These are also things that can happen in microbial communities, especially the kinds that are sort of in you and me, and in particular in our guts, which is that even in our gut microbial communities, you can see these transitions due to specific perturbations. And so let me show you an example of what this kind of data looks like. So here you sort of see a plot where the y-axis shows you the fractional abundance. So this is kind of like the frequency of sort of different bacterial species. So each color over here is a different bacterial species. And on the x-axis, you just see time. So this is one person being tracked over several days, and so their gut microbiome is being sampled. And so you can kind of see that, up until this day, 150 or so, these different bacterial abundances, they're kind of fluctuating. Maybe they're changing a little bit, but maybe one can reasonably say that they're in, some kind of stable state. Now, at about 150 a day, what happens is that this person gets a specific infection, and so in particular, you can kind of see that this little fluorescent green species, which is actually the species that infected this person, starts rising up in abundance. Eventually, this infection starts getting cleared, so the species starts going down again. But once the infection sort of clears out, which is the end of this gray area, you can see that the community kind of ended up in sort of a different state, right? So this is kind of what I marked out in state two. Right. And so the idea again is that if you didn't see the infection per se, on the left side of this, you see one sort of state, and on the right hand of this, you see maybe another sort of state. And again, the infection can act as sort of perturbation to transition communities between these different states. And this is kind of a fairly common phenomena that you see in gut microbial communities and lots of other microbial communities as well. And so really the question that we were asking, or the phenomenon that we were grappling when we were doing this project, was the fact that you can see these multiple stable states in microbial communities appear often in very similar environmental conditions. So, just to see in this cartoon, the x-axis is some sort of environmental axis. The y-axis is some sort of community state axis, and you could see basically communities being able to transition from state one to state two. And the idea is that basically what can distinguish state one from state two is there are different species at different sort of fractional abundances. And so the question that we were asking was, what can explain this kind of observed multistability? Were there simple models to understand this? And presumably, if we had these simple models, maybe they could tell us something about what kind of perturbations one could use to be able to transition from one state to the other. Remember, in the gut, this is quite important because one stable state might sort of correspond to good health, and one might correspond to somewhat bad health. And so this can be quite an important thing to sort of learn about. And so we found that indeed there is a very simple model that could explain this observed multistability. But this answer sort of came from the idea that basically lots of species in microbial communities often compete for the same set of nutrients. And we could explain multistability if we sort of focus on the idea that microbes often use one nutrient at a time. So let me now explain to you what is it exactly that I mean. Like I just said, this is often true, and let me try and convince you very quickly that multi-species communities, especially in microbes, often compete for the same set of nutrients. So again, this is sort of a cartoonized version of some pretty impressive data collected from Eric Martens's group in 2017. And so what you see here is basically a graph where on the sort of bottom row are basically different microbes, and these are really different microbial species of the phylum Bacteroides. And for the purposes of the cartoon, different colors are different species. And on the top over here, you see different nutrients, in particular, different polysaccharides that are often present in the human diet. And basically, connections in this graph tell you if or if not a particular species, for instance, this blue, is able to eat this particular polysaccharide. So, for instance, a link from this blue to the CS, which is chondroitin sulfate or something, indicates that basically this blue species has the capability of consuming chondroitin sulfate. And so you can pretty easily see that this is both a very dense network, right? Which is to say that the same nutrients can be consumed often by multiple species. And also that basically, each species is pretty sort of prolific in being able to eat multiple nutrients, right? So it's true that different species can eat many different nutrients, and also that the same nutrients are being competed for often in between sort of many species, right? And often it's also true that in these gut microbial species, when species are presented with this sort of buffet of many, many nutrients, it's not true that they eat all of them at the same time. Often, microbes typically eat nutrients one at a time, and often they show what's called a resource preference hierarchy. And so the idea for this goes back to the classic days of Jacques Monod doing his thesis in the 1940s. And this is sort of like again a plot which sort of condenses the information from what he discovered, where the idea is that if you grow bacteria, in this particular case E. coli, in a medium containing, a mixture of two different sugars, for instance, in this case, glucose and lactose, what you can often make are these very nice growth curves. So on the y-axis, you can plot some measure of the population size, in this case, optical density. And on the x-axis, you can just plot times, right? So really, this sort of a red curve over here tracks the bacterial population size. And so you can see that basically bacteria initially have a lag, then this population starts growing exponentially. Eventually, this population stops growing and enters a somewhat static situation, after which it starts growing again in a second phase and then finally saturates. And so this biphasic growth is really the one that I want you to focus on, because if you sort of look underlying the growth of these bacterial populations at what the nutrient concentrations look like, well, you can see something pretty clear, which is that in the beginning, in the first phase of growth, it's actually only glucose whose concentration is going down. And in fact, the concentration of lactose is pretty rock solid. It's not really changing. And it's really during the second phase of growth that the lactose concentration is going down. Right? And so the idea this sort of brought about, and this is it's sort of well accepted idea today, and it's called dioxy, is that often bacterial growth occurs in two phases. First, bacteria, in fact, E. coli, in this case, use glucose and not use lactose. After this, after they're done sort of consuming the glucose, they'll have this sort of phase during which they're rewiring their internal metabolism and getting ready to use lactose. They'll then use lactose and grow on it. And then, of course, there's nothing left in the medium, so they'll stop growing. And so this kind of dioxic growth, we know now, is sort of these kinds of preference hierarchies. This idea that you use glucose first and lactose second is really sort of encoded in the regulatory networks of bacteria. And because these regulatory networks can often be very plastic, we now know from studying things that are different from E. coli that these can be different for different microbial species. So in fact, while E. coli might prefer glucose over lactose, some other species might actually in fact prefer lactose over glucose. And it's also quite sort of generalizable. So for instance, it's also true that many of the gut microbial species that I talked about in the previous slide are also dioxic for sort of many of these polysaccharides that I was mentioning. And so we're kind of all set, right? So we have this idea now that microbes could have preference hierarchies for several nutrients. And then, of course, if microbes sort of grow on different nutrients, they could be differently competitive on them. And so this is really the idea that we exploited in our sort of model. And so really all we need now to get this sort of model in place, which I'll describe to you now, is two ranked tables. So the first ranked tables just tells you which microbes prefer which nutrients. And so the cartoonized version of this is here. Let's say there are two microbes, a red one, M1, and a sort of yellow one, M2, and three different nutrients, N1, N2, and N3. You can sort of chart out, by doing experiments or by looking at genomes, which nutrients these microbes prefer most to which they prefer least. So in this case, I've sketched out a cartoon in which M1 prefers N1 the most, N2 the second, and N3 the third, while M2 prefers N3 the most, N1 the second, and N2 the third, right? So you can also condense this cartoon in this kind of table, and that's what I mean by the ranked table over in my title, which is that basically you can condense the same information where each of the different species are in rows, and the ranks are the ranks of the different nutrients are over here on the columns. So that's about the preference hierarchies. You can also now ask for each of these nutrients which species is more competitive, which is that if you only had that nutrient available and both these species growing in sort of a medium, you often see this phenomenon called the competitive exclusion principle play out, where really one of the species will grow a little bit faster than the other on these nutrients, and so will win the competition. And so you can also chart that out in sort of a second rank table. So for instance, you can say that for nutrient one, the sort of yellow bug wins over the red bug, for N2 the red over the yellow, and finally for N3 maybe the yellow over the red. And you can also condense this stuff into competitive abilities of microbes, where you have different nutrients and different ranks, where rank tells you, well, this is the most competitive species on N1, this is the second most competitive species on N1. Right? And so once you have these two ranked tables, you can really condense all the information about microbes that you need to know. But remember, what's our goal? Our goal is to be able to describe something about multiple stable states that these microbial communities might have, right? And so you might be a little bit confused right now. Where is that going to come in? And so really the idea is that the inspiration for thinking about multiple stable states in this framework comes from what's called the stable marriage problem, which is kind of like a famous problem in game theory. And so let me kind of explain to you what I mean here. And of course, please excuse the sort of archaic heteronormativity that lies ahead. But the idea is, let's maybe transport ourselves back into the sort of Victorian age where, if you are familiar with Pride and Prejudice, then maybe these characters are a little bit familiar to you. If you're not, then there are four men and four women, and they all go together to, let's say, some sort of Victorian ball. And in this ball, they all decide that, oh, we would actually like to get married to each other and paired up. And now the question really is, how should we do that? So they all come up with this idea, which is that, okay, each of the men and each of the women can rank each other based on their preferences. So, for instance, each of the women, for instance, Elizabeth here has sort of a ranked list for which men she would like to get married to. So, for instance, Elizabeth on her list would like to get married to Wickham the most and Collins the least. And all the women do this, all of the men do this. And now the question is, once you have these sort of preference lists, they want to get paired up. And so, you might make some random set of pairings between these men and women. And so the question in the stable marriage problem really is how do you tell if a given pairing is stable or not? And so let me just describe what the notion of stability in this sort of problem is. The idea is that often, like random pairings or randomly putting people together can result in what are called eloping pairs or blocking pairs, and which are often unstable. And so the idea of instability is that if Collins and Elizabeth were paired together and Darcy and Lydia were paired together, for instance, this would be an example of an eloping match or a blocking match, or in other words, an unstable match. And the reason for that is that Elizabeth and Darcy, who are both not partnered with each other, currently like each other better than the partners that they have been paired up with in this particular pairing. And so the idea is that because they both like them each other more than their current partners, they would kind of elope away. That would block off this sort of pairing, and this whole pairing would become unstable. Right. And so really the idea in this sort of framework is that stable pairings are those specific non-random configurations where no one can elope, or so where it's sort of all pairs are such that you can take no man in one pair and no woman in another pair and find that they both would like to get married to each other more than their current partners, right? So this is really a coincidence matching. You really want both to be the case. And so really, it is sort of Gale and Shapley that showed that basically for any set of pairings that you can construct between men and women, there always exists one, there always exists at least one stable pairing. So there's at least one stable matching. And in principle, there are more, and they sort of prescribed algorithms for how to find these pairings that are all stable. And so their idea for sort of I'll give you sort of a brief idea for what this sort of algorithm was. So basically, the idea is very simple. There are sort of two versions that are symmetric. So one of them is called the men proposing algorithm, and one of them is called the women proposing algorithm. And this particular example, I believe you see the women proposing algorithm, which is basically on the first day, basically all of the women go to the first man on their preference list and they make proposals. So in this case, so for instance, Bingley received 3 proposals, Wickham received 1. And so basically, every man who receives only 1 proposal must say yes, and every man who received multiple proposals must only say yes to the woman who was on the top on his preference list from the proposals that he received, and sort of reject the others. So in this case, that turns out to be Jane, and Lydia and Charlotte have their proposals be rejected. So then on the next day, they kind of go down their preference list. They go to the sort of the second man on their preference list. And so, this kind of can go on and on and on. And this sort of continues until you sort of stop, until every man is paired with one woman. And in this case, you find this particular set of pairings. And basically, Gale and Shapley proved that this pairing is stable. And so, and maybe if you sit down for a while on your own, you can sort of figure out exactly why this pairing is stable. And I'm happy to sort of explain that to you off the top, basically. But the idea really here is the following, that you can really draw an analogy between this idea of matching men and women and have their pairings be stable to the idea of microbes and nutrients and have their pairings be stable, which is that just like there was this idea of ranked preference lists for men and for women, we also have this in this construction of the problem that I was describing to you, which is that for microbes, we have different preferences for nutrients, right? So you might think, okay, that makes sense. The active party is microbes, just like in the previous example, the active party were women. But, very important in this algorithm was sort of men making choices based on the proposals that they received. And of course, nutrients are not very active or very sort of, sentient, as you might have guessed. But the idea really is here is that the nutrients are effectively making a choice when two microbes are sort of trying to compete for them because of the competitive abilities of these different microbes. So effectively, if both yellow and red microbe here go for nutrient one, it's really the competitive outcome of yellow beating the red that will appear like basically the nutrient one making a choice of yellow over red. And so really, once you have these preference tables, you can really sort of, deploy the edifice of the stable marriage problem. And sort of, because this analogy seems exact in this particular limit, you can then ask questions about what the kinds of stable states are that you expect to get. And so the only thing you really need to deploy this, like I said again, are these two sorts of ranked tables. So here I'm showing you sort of a more complicated example, just in the interest of time, which I've sketched out for sort of 7 microbial species on 7 nutrients. So this is actually a very similar and inspired to the original example of these gut microbial species that I was showing you. That's why the colors are sort of designed to be very similar. And so in this case, say that we have these kinds of, preference hierarchies of the different microbes for the different nutrients, and also the competitive abilities of the different microbes for the different nutrients. Well, then once you have this table, that's all you need to be able to basically enumerate all the stable states. So here's one example of what these stable states looks like. So in this particular stable state, which I've marked out as S1, you have this green bug using nutrient 1, you have the purple bug using nutrient 2, the blue bug using nutrient 3, and so on and so forth. So you basically have a one-to-one matching from every nutrient to a microbial species sort of using that nutrient. And so this is in particular, stable state 1, but in principle, for this particular table that I'm showing you on the left, it turns out that there are 5 other there are five total stable states, and those are one in each column that you see over here. And so, in some of the stable states like S1 and S2, this green bug is using nutrient one, but yet in others, you see the red bug is using nutrient one. And so, really, I want you to take away sort of two things from this sort of slide. One is that once we have these tables, we really can enumerate all of these stable states by going through these kinds of algorithms that I was mentioning. And B, the number of stable states that you end up with, which is here five, is actually a much smaller set of the total number of pairings that you could get. So remember, for seven species, you really could get 7 factorial possible pairings, right? Which is much larger than five. And so it's really true that most pairings that you will have for nutrients and microbes will actually be unstable. And so finding these sort of small set of stable states is really sort of what was the goal that we sort of came up with at the sort of beginning of this talk. And if we have this sort of ranked framework, this ranked preference framework, we can do it. And so really the idea is that once we have these stable states, you can also then talk about transitioning from one stable state to another. So here's an example of what you can do if you want to transition from state 2 to state 4 in the previous slide. And really the idea is that the way you achieve a transitions from one state to another is by doing what's called a transient nutrient removal. And so basically the idea is, let's say I start off with state 2, which is shown over here in this first column, and I transiently remove nutrient 1. Well, then in that case, what will happen is that the species, this green species using nutrient 1, will get orphaned transiently. And so it will want to go down its preference list. So it can go, let's say, to nutrient 7, which if you look at the previous slides table, that is indeed what this green species preferred next. It will then try to then, of course, compete with the red species for nutrient 7. And if it's more competitive, which in this case it is, it'll knock out the red species. This red species will now go down its preference list from nutrient 7, which in this case turns out to be, for the purposes of this example, nutrient 1. And then at this moment, if you reintroduce nutrient 1, which is that if this removal was transient, then this red species can establish on this nutrient 1. And actually, what you'll find out is that you end up with indeed a new stable state, state S4, which is again stable. So again, if you take any two sort of microbes and try to see if they prefer nutrients to each other, they'll not be able to do so. And so indeed, you sort of started off with sort of one stable state, and by finding a very specific perturbation, in this case, the transient removal of nutrient 1, you can end up in another stable state, this state 4. And indeed, you can do this for sort of many pairs. And if you do it for sort of all the different pairs of states, you can sort of end up with this sort of state transition diagram or the state transition graph that you have here on the right. And so basically the way to read this graph is that the different nodes here are the different states, right? These five different stable states that I was pointing out. And basically an arrow from one state to the other tells you whether it's possible to go from one state to the other by a direct perturbation or not. And so, for instance, from S2 to S4, you can do so by transiently removing nutrient 1. And so that's why you have N1 sitting over on this edge. And the other thing that you'll notice is that these edges are all directed, right? Which is that it's possible to go from S2 to S4 and S3 to S4, but, for instance, not from S1 to S4, or directly at least, or S4 to S3. Right? And these sort of like self loops over here just sort of indicate that most perturbations when you're sitting in S2 and actually just bring you back, right? So they kind of indicate the resilience of each of these states. And so if you sort of look at this state transition diagram, you'll see that it's actually very hierarchical, right? Which is that there's a state S1, which you can start off with. But then if you make perturbations, then you sort of go down and then you go down again and then you go down again in a way that's kind of irreversible for you to go back, right? And so this is by perturbing nutrients. And so in this way, we kind of achieve the thing that we wanted to achieve, right? So we kind of wanted to, we sort of set out with this idea of wanting to have kind of a simple model in which we could explain sort of multiple stable states and sort of understand how to transition between these different states. And so what we found was that in this particular case, if it is indeed true that you find that microbes are consuming nutrients sequentially or one by one, then sort of by making this mapping to the stable managed problem, you can do so. You can do so exactly. And so with that, I will end my talk, and I'm happy to take questions.
Questions?
Hello, can you hear me?
Yes, yes, I can. Hi there.
I was wondering, did you test this experimentally?
That is a good question. Maybe I'll ask you a question what exactly would you mean by testing this experimentally? You mean whether we could see the stable states as predicted by the model?
Yes, I imagine perhaps measuring the nutrients would be difficult, but you
Yes.
might see, like a change in composition that goes over across multiple deoxic shifts.
Right. Yeah. Basically, I think the issue with sort of testing this out in more complex communities is exactly sort of like figuring out all of these hierarchies for different species. Turns out to not be such an easy challenge. So initially, we were very sort of gung ho about, going to experimentalists and getting them to do these things. But it turned out that actually figuring out these hierarchies for, more than a few different sugars turned out to be a pain. And so really what I'll say is that for sort of communities with a small number of species, maybe two or three, we indeed found that the kinds of stable states that we can predict are the ones that you often see. But really the magic of this is to do it in more complex communities. And so we're still sort of looking out for the technology in some way to catch up for us to be able to test these things out.
Right. Thank you. It's very cool. Bye.
Thanks.
Okay. So, can you hear me?
Yes.
Hope so. So the marriage problem has a very nice structure, of course.
Mm-hmm.
The stable states have a very nice structure. And
Yeah,
transition paths between stable states have a very nice structure.
that's right.
Enabled you, I mean, if only theoretically perhaps, to play with these systems to get it to move in a direction you would like it to move. Perhaps going back to your original motivating example, right? You may wish
Yeah.
from one stable state to another.
Yeah. Yeah, so sorry, just so because it's a little bit hard to catch everything with the mic, maybe I can repeat the question and you can tell me if I sort of got it right. Is the question that because of the nice structure of the stable states, you can say, can you say something about what kinds of states you can go to from which others?
Well, I mean, I know I can say that, but
Yeah,
I was wondering, given the nice structure
yeah.
that we have on these problems, does that create some, I mean, it would seem to create some way that you could say, well, okay, we're at a stable state that we don't like. Here's how we can change the system to get somewhere better.
Yeah, absolutely. Yeah, that's exactly the idea. The idea is exactly this, that if in sort of more if you have a real community that is in some state that you don't like and you would like to transition it to another state, if you knew something about how the different microbes in that community were able to use nutrients, then really by building these kinds of graphs, you can know exactly what kind of perturbations to make in the lab to be able to make in nature or the lab in per se, to be able to transition out of those states. And right, so the main thing about the structure of these problems is that most perturbations don't take you away from the state, right? So it's actually very directed perturbations that do that. Yeah, so that's indeed the goal, to figure out exactly what those perturbations are that take you away from sort of undesirable states and towards desirable states. Does that answer your question?
It answers my question. It's I still have a lot more questions, but I'll have to search the manuscript and
Yes, and yeah, please feel free to write to me. I will be happy to chat more.
Right, lovely. Thank you. Thank you very much.
Thanks.
Hey, can you hear me all
Hi.
right? Well, thanks. This was super fun. I just wanted to ask about how you're thinking of this model as like explaining the target phenomena. Because I mean, the model you're showing here has an absorbing state, but you sort of started off with like gut bacteria, which would make transitions. I mean, I see that the bacterial bloom case maybe is not that transition. Maybe it goes one way, you're arguing.
Yeah.
But yeah, so like, would most of these examples have absorbing states, or would they have cycles, or yeah?
Yeah, excellent question. So let me just whip out one of my backup slides. So this is actually a wonderful question because this is also one that we were concerned about. And so the answer turns out to be that no, you don't have absorbing states in this system because just like there's a transition graph for perturbing nutrients that I showed you, in the interest of time, but I didn't show you that there's actually a mirror world. That's sort of this mirror graph over here on the right, which is sort of what you get if you make transient perturbations to microbes, which you might do, let's say, by administering specific kinds of antibiotics. And in this case, actually, what you can find is a whole other world of sort of perturbations that will give you the exact mirror opposite of this graph on the right. So you can, in fact, you could go from S1 to S5 by hitching a ride on these nutrients, but then you can go back from S5 to S1 by sort of transiently perturbing specific microbes. And so it's so, you can actually go from any to any in principle if you're able to perform perturbations of both kinds. And so indeed, it might be possible, in the sort of cyanobacterial case, like you were pointing out, it might literally not be possible to perform some of these perturbations on the right. For instance, how do you kill like a bajillion cyanobacteria? But in other cases, especially in the gut, we believe that some of the, like both of these kinds of perturbations, or at least a combination of them, would be possible, which would allow you to kind of cycle between these states in a way that you would want.
Dr. Akshit, thank you for the really nice talk. So I was wondering, I'm not sure about this, but still, the key assumption for this work is that bacteria will have like a stable preference order for these kind of metabolites or nutrients, let's say. But I'm not sure. So aren't there cases where, based on the environmental conditions, the bacteria would change their preference order? And if throughout the process that we are modeling, like the stable state during these transitions, if these preference orders are not stable for either all the bacteria or even one of the bacteria, then what would be the implications for approaching it in this manner?
Okay, great. Nice question. So I would say a few things about the sort of plasticity of these orders. So one of them is, of course, that the preference orders can change, but usually they change sort of over sort of evolutionary timescales. And so what you'd see is that maybe different strains of bacteria can change their preference orders over sort of, at least the cases that I'm familiar with, is sort of several hundreds to thousands of sort of generations. And so in that case, because this process of ecological assembly, where you get these kinds of communities, because that's much faster than the rate at which these regulatory networks can change, I do believe that basically that doesn't quite affect these kinds of stable states that I'm talking about. But sort of even if you were to change these kinds of preference orders over sort of evolutionary time, well, then you'll just go from sort of one landscape of stable states to another landscape of stable states, right? This sort of process of ecological assembly is a pretty sort of rapid one.
So maybe I can ask one.
Yeah, sure.
Akshit, Sagar here. So building on the question, I'm going to ask an outrageous question.
Okay, go ahead. I love those.
If there is preferential ordering, even at evolutionary timescale and all that, could it be that the 4 axioms of von Neumann utility theory for the order preference is also satisfied by these bacteria, and we can call them rational, VNM rational?
I wouldn't go so far as to do that. I think that I think that really what these bacteria are doing is sort of really. I mean, if you actually dig into how exactly these choices are being made, I mean, we actually don't fully understand what couples, bacteria having the preference hierarchies the way that they have. So, and often what you see is that the preference hierarchies that are encoded in bacteria are sort of far from rational. So, for instance, even in the case of E. coli that I was sort of mentioning when I was giving an example for glucose and lactose, what you might expect in life is that because E. coli prefers glucose first over lactose, it would grow faster on glucose over lactose. But actually, this is not true. What is often true is that this E. coli bug is sort of anomalous and non-rational in the sense that it actually grows slower on its first preference and goes faster on its second preference, which is kind of weird and crazy. So actually, we have like a whole bunch of ideas for how you might explain this kind of irrational behavior, which has to do with the fact that these microbes are not growing alone, but they're often growing in communities, which can change, which can actually change what the rational thing to do is. But I think that to actually make that more concrete would require a little bit more than just von Neumann's postulates.
I was sort of asking this also because there are these experiments on fish and
Yeah.
flies and insects where
Yeah.
they check this rationality, but
Mhm.
by maybe diluting the preference, like statistical preference ordering and all that, right? And so there are certain
I see.
papers where they claim the fish is rational because the preferential ordering they are following transitivity and all that. So yeah,
I
was
see.
just thinking whether it could be checked on bacteria as well.
Yeah, okay. That's a good point. So I'm not too familiar with these fish experiments that you're talking about. I would love to sort of know more about them, and maybe I can write to you after. But as far as I would sort of go on to say is that, I mean, often bacteria do things that don't seem to be rational. So I wouldn't necessarily buy this in the first place. But I mean, who knows? I think my take on this is that they're probably doing something very rational or on a very different level that we don't quite comprehend, which is this sort of time scale of like how exactly they survive in communities which are changing in sort of all sorts of fluctuating ways. So I think in this case we might not possess the right tools to be able to say what rational behavior is because of the complexity of communities.
Thanks, Akshit, for this really fascinating talk. So
Thanks a
next
lot.
time we hope to have you here.
Yeah, same here.